I'm Dr Merry Wilkinson. I'm doing a postdoctoral research fellow under Dr Simon Eatons and Professor Lucy Wedderburn. The project I'm going to talk about is the Pump Prime project that was awarded to Dr Simon Eatons back in 2018. It was looking at defining mitochondrial dysfunction in juvenile dermatomyositis with the focus to look towards new treatment target. The project is looking at juvenile dermatomyositis, or JDM, from pre-treatment and on-treatment patients, so pre is at baseline, and comparing this to age-matched healthy control samples. With those samples, we looked at monocytes, and what we've found is there's increased mitochondrial superoxide, that we have increased oxidized mitochondrial DNA within these cells. Looking at metabolism, we've, through an assay where we showing that there's decreased lactate production, and this methodology is also looking at other such things, such as the production of carbon dioxide and elements of the pentose phosphate pathway. As a potential biomarker for this disease of mitochondrial dysfunction, we've looked at mitochondrial DNA in plasma, which we've shown that there's an increase of this cell-free mtDNA. Functionally, we've looked at oxidized mitochondrial DNA and whether it induces interferon, and the readout is by PCR gene expression of specific genes in the interferon 1 pathway. And we've shown that there is an upregulation by oxidized mitochondrial DNA. We've also looked at seeing whether drugs could target and downregulate this interferon expression, and we've shown that TLR9 antagonists and N-acetylcysteine can block this response. And we're trying at the moment to translate this to see whether these drugs could potentially downregulate interferon in the disease itself. As outputs from this project, I've developed two methodologies. One is to see if we can measure oxidized mitochondrial DNA in monocytes. So taking monocytes, I've isolated the mitochondria and then precipitated out the mitochondrial DNA. By dot blot Western, we've managed to quantify the amount of oxidized mitochondrial DNA using 8-OHDG, which is non-specific, but because we've isolated the mitochondrial DNA, we can confirm that this is oxidized mitochondrial DNA. The second methodology that I've created was generating oxidized mitochondrial DNA as a reagent. So I took genomic DNA, and by long-chain PCR, I amplified the mitochondrial genome in 2 fragments. I then amplified this mitochondrial DNA and oxidized it using chromium trichloride, creating this reagent, which I then used in the functional assays. In terms of COVID-19, this has affected my access to the lab, so my ability to continue with these experiments, but have very much tried since June to increase my output. It's quite difficult ordering reagents at the moment. For example, PCR plates I ordered almost a year ago arrived in December, which is quite a long time to wait for these specific plates, but other things are taking quite a long time. One of the key things is patient recruitment. We have got a few patients, but the most problematic is getting healthy control samples at the moment. See, adapting to working from home, which is quite different from being in the lab, was sort of volunteering, so supported the clinical labs with the COVID testing. Seems a very long time ago now. In terms of further funding from this initial grant, which was between 2018 and 2019, I applied for several fellowships, but I was successful in getting a 3-year fellowship from CureJM, which is supported by the GOSH BRC, so I'm very grateful. And this is supervised by Professor Lucy Wedderburn and Dr Simon Eatons and Claire Deacon, which, as Dauda said, she's now returned to Australia. So this fellowship expands on the aims and findings of the project discussed, and this is defining mitochondrial dysfunction in JDM and to, again, investigate novel treatment targets. And this is, I generate these experiments over the Wedderburn and the Eaton research labs. So the outputs from the project so far In 2019, I gave a talk at the Arthritis versus Arthritis Centre for Adolescent Rheumatology Symposium, of which I was actually awarded a prize. I then went on to give a talk and received a fellowship to attend the GCOM conference in Berlin. And then in December 2019, I presented a poster on this work at the British Society of Immunology Congress in Liverpool. I then went on to attend the virtual conference for press in September 2020, where I gave a lightning talk and presented a poster. And then more recently, I was invited to give a half an hour talk at the 5th versus Arthritis Centre for Adolescent Rheumatology. So thank you. I can do this project without all of these people and the funders. So this is how we get these things done. But thank you.